Hello, I am Quinn Girasek, a graduate student at the Virginia Institute of Marine Science. I use molecular techniques to study billfish. For those of you who are unfamiliar with billfish, this is an umbrella term used to categorize swordfish, sailfish, marlin, and spearfish. One part of my research involves a diet comparison between the white marlin and round-scale spearfish. This lesson incorporates some of the methods I use and serves as an introduction to diet analyses and bioinformatics. The activity was created with high school's biology standards in mind, but it can be scaled up for AP classes and highly motivated or advanced students with the extension activities in the second worksheet. Students will learn about food webs, diet analyses, and how to use bioinformatic resources. In this lesson, students will collect data and learn how to identify organisms based on DNA sequences. The lesson has 3 parts. The background will be given by the instructor using the provided PowerPoint slides. This will provide the necessary information to complete the lesson and to introduce the lesson itself. After the background will be the activity. When I'm out in the field collecting data, it is difficult to identify digested species. Sometimes there is only liquid, and other times the stomach contents are mostly digested, so it is difficult to identify the fish or invertebrate. The activity will allow students to search for and identify organisms using DNA sequences. Students will draw fish bones out of a paper bag and put together the bones. When done correctly, a species DNA sequence will be complete. Note that although all of these are fish bones, some of the sequences are not fish. Students will work in groups and use NCBI BLAST to answer questions on the provided worksheet. If time allows, students will create and infer phylogenetic trees from the organisms they identified. The post-activity is a discussion which will allow further understanding of the concepts presented in this activity. Through this lesson, I hope that you and your students become interested in billfish and genetics. If you have any questions, don't hesitate to reach out. Thank you for watching.